Good morning children. In today's class, we shall discuss in detail about inheritance. Okay, inheritance is one of the basic concepts in genetics. So what is inheritance? Inheritance is the phenomenon by which some of the characters of parents are being passed over to the next generation. Okay, in those days they were thinking that some factors in this organism is being transmitted or transferred to the next generation. Now we know that these factors are genes. Very good. So in mid 19th century, when Mendel did the experiment on pea plant, he observed 7 different contrasting characters. Okay, like tall and dwarf plants, round seed plants and wrinkled seed plants. He also observed plants showing yellow color seeds and green color seeds and so on. Okay, 7 different contrasting characters. And when he was studying, he first observed only one contrasting characters and the remaining characters were almost similar in the 2 sets of pure breeding plants. When I say pure breeding plants, what do I mean? It means that particular plant for generation after generation were showing only same character. Okay, there was no deviation, there was no variation. Okay, that is why we call that as pure breeding or pure line plants. So he took a pure breeding tall plant and he also took a pure breeding dwarf plant. Okay, so that means all the other characters are similar excepting for this one character. Okay, that is why this cross is called monohybrid cross, because he is only taking a plant with only one different contrasting character. Okay, or I can say trait, different character or different trait. Okay, so now let us consider this tall plant and the dwarf plant, okay, as parents. What are they? Parents. Okay, so this is the parent generation. So pure breeding tall plant, pure breeding dwarf plant. These two are parental generation. Okay, so if you have to write the genotype of the tall plant, I will write it as capital T, capital T. If I have to write the genotype of dwarf plant, I will write small t, or I can also say lowercase t. Okay, lowercase t, lowercase t. Now, how will these plant produce the gametes? See, we all know during the gametogenesis, when gametes are formed, diploid cell will become haploid gametes, isn't it? So, each gamete will have only one allele. Now, what is an allele? Yes, so a gene may have alternative forms. Okay, so here in this case, capital T is a gene and the alternative form of capital T is small t. So capital T is one allele, small t is another allele. Okay, so in this capital T, okay, in tall plant, you can see that both the alleles, okay, are similar. So I call this as homozygous. How do you call it? Homozygous. Very good. In this dwarf plant, how are the alleles? You have small t, that means both are similar. So I call this as homozygous. Very good. So when gametes are formed, you will have capital T gamete and capital T gamete with respect to tall plant. In case of dwarf plant, you will have small t gamete and small t gamete. Are you clear, children? All the gametes produced from the tall plant will have one capital T. Got it? All the gametes produced from the dwarf plant will have one small t. Are you all clear, children? Now what he did was, he emasculated the dwarf plant. So what he did was, he removed the stamens. So once you remove the stamens, can there be any self-pollination? No. So he artificially, okay, by force, he removed the stamens and dusted the pollen grains from the tall plant. So what is he actually doing? He is cross-pollinating. He is cross-pollinating the tall plant and the dwarf plant. So in the next generation, we call that as filial one generation. That means F1 generation. So in the F1 generation, to his surprise, he found that all the plants were tall. All the plants were tall. So what did he do? He allowed them for selfing. So he allowed them for self-pollination. In the F1 generation, you can see all the plants are tall. So he allowed for self-pollination. Then the next generation, that is the filial 2 generation, F2 generation, he found that some of the plants were dwarf. Some of the plants were dwarf. Now he started analyzing. Okay, what is this? Some characters are hidden in one generation, but it is again expressed in the next generation. So what is happening here? So when he was analyzing the data, he did repeated experiments. Okay, not only with tall and dwarf. He was doing it with round and wrinkle. He was doing it with green and yellow. Okay, like that he was doing with only one character. Okay, so all the results of monohybrid cross were similar. So he found that in the F1 generation, the plant was heterozygous. Okay, that means though the plants are tall, if you see the genotype, it was capital T and small t. Capital T and small t. And during the gamete formation, what will happen? Yes, during the gamete formation, 50% of the gametes will have capital T and 50% of the gametes will have small t. So what would be the combination? Here comes our Punnett square. Okay, so Reginald C. Punnet, he devised this, graphical representation of genotypic ratio. So he gave an idea, okay, how to find out the combination of any genotype when you are doing a cross. Okay, so you can write the male gamete on the top and get the female gamete on the sides. Okay. So if you just cross, you can see capital T and small t will segregate into capital T and small t. So one gamete will have capital T, another gamete will have small t. So as I told you already earlier, 50% will have capital T, 50% will have small t. So now I am taking two F1 generation plants which are heterozygous, tall. So where they will be have 50% capital T and 50% small t in their gametes, isn't it? Now see, when you are using the Punnett square and finding out the combination, here I told you capital T with capital T will have homozygous tall, capital T with small t have heterozygous tall, small t capital T you have heterozygous tall, again small t you have homozygous dwarf. Okay, so out of 4 plants, 3 were tall and 1 was dwarf. Okay, so the phenotypic ratio of monohybrid cross is 3:1. 3 tall, 1 dwarf. What is the genetic makeup? Were all the plants showing similar genetic makeup? No. See, here you have 1 homozygous tall, 2 heterozygous tall and 1 homozygous recessive. Therefore, the phenotypic ratio of monohybrid cross is 1:2:1. Are you clear, ma? So here, he brought into the first and second law of inheritance. What is this first law? Very simple. See, he said, the characters or traits which are expressed in the first generation, that is first filial generation or F1 generation, are called dominant characters. So which one is expressed here in this case, ma'am? Tall. Very good. So tall is a dominant character. Which allele is dominant? Capital T. Very good. But in spite of having a dominant gene, in the next generation on selfing, when the small t and small t, okay, the recessive alleles were given a chance to combine together and express, they are expressing their character as dwarf, isn't it? So those genes, okay, those allelic genes, when they are given a chance to become homozygous and when they are able to express themselves, such characters are called recessive character. What do you call them? Recessive character. So in the first filial generation, the character which is expressed is called dominant character and that which is not expressed is called recessive character, which will be expressed if given a chance to combine with another recessive allele in future generations. Clear, children? This is the first law of inheritance. What is the second law? The second law is law of segregation. So when I say segregation, that means here, see the capital T and small t, both the alleles were together in the hybrid, isn't it? F1 hybrid, both the alleles were together. But during the formation of gametes, they separated. Were there any mixing? Were there any confusion? No. Both the alleles, they separated as individual capital T and individual small t. So this is called law of segregation. So during the gamete formation, though there are two alleles in an allelic pair, the alleles will separate without any overlapping. They will separate and each allele will go into a specific gamete. Are you clear, children? This is with respect to monohybrid cross. So here we have learned about inheritance of one gene, one single gene. Okay, and we also know gene has different alternative forms which we call them as alleles. Okay, we call them alleles. And genes were earlier called factors. Clear, children? Shall we move on to dihybrid cross? See, having learnt the monohybrid cross in detail with the basic principles, okay, what is an allele, what is an allelic pair, what is homozygous, what is heterozygous, yeah, this dihybrid cross becomes so easy, okay. So here, what is the difference? He is taking two plants, okay, two pure breeding plants with two contrasting characters. Very good. So what are the contrasting characters Mendel took here? He took one pure breeding plant with yellow and round seed. He took another plant with green and wrinkled seed. Okay. So now what did he do? He took the pure breeding round yellow and he took pure breeding wrinkled green. So when I say pure breeding, that means they were only showing these characters for generation after generation. Clear, children? Now see, for round we are taking capital R, capital R. For yellow we are taking capital Y, capital Y. So similar to that, we are going to use same alphabet but in lower case for the recessive character. Okay. So capital R there means we are going to use small r here and small y here. Clear? Small r means it is wrinkled. Small y means it is green. Clear? So now the gametes. How will the gametes be? When the gametes are formed, just now we saw, isn't it? They will segregate. How do they segregate? Yeah. One capital R, one capital Y. Now, during the first filial generation, F1 generation, what we are doing? We are cross-pollinating. Very good. Similar to that story, what we are doing here? We are remaxulating. Okay, we are removing the stamens from one plant and dusting the pollen from the another plant. So now here, what will be your F1 generation? Very good. Dominant character will be expressed. What is the dominant character here? Round yellow. So all the plants will be producing round and yellow seeds. Very good. So now what are we going to do? We are going to let them self-pollinate. Okay, so on selfing, you can see how the next generation will be. Okay, next generation. So again, I told you in Punnett square, you can find out the various combination of your genetic cross, isn't it? So now, which way you are going to write? You can write, okay, male gametes in one side, female gametes in another side, and you can cross it. You can just cross it, and as you write, okay, you will understand that you find, see there is a triangle here, okay, like this. You can draw a triangle like this. Okay, so you can find 9 of them showing round yellow seeds and 3 of them showing yellow but wrinkled seeds. 3 of them showing green but round seeds. 1 showing your green wrinkled seeds. Okay, so this is how he got the dihybrid cross. He got the dihybrid cross. So what is the phenotypic ratio of dihybrid cross? It is 9:3:3:1. Okay, children. So now, here it is very clear that one allelic pair will not influence on the other allelic pair during this segregation, or this is called law of independent assortment. So if capital R is going to be in one gamete, it will not say, come on, small y, don't come near me. Only capital Y has to come. It will not say that. Because this allelic pair will assort independent of the another allelic pair. So capital R and small y can be together. Small r and capital Y can be together. Do you understand? So each allelic pair will assort independently without the influence of the another allelic pair. This law is called law of independent assortment. Clear? Third law of inheritance. So today we discussed about the inheritance. Okay, what is inheritance? What is monohybrid cross? How we are deriving at the ratio phenotypic and genotypic ratio using Punnett square. And we also discussed about what is a dihybrid cross, and we discussed about the law of independent assortment. Okay, children? So I think this is very clear to you. If you have any doubts, you can come back to me, you can call me and clear your doubts. Thank you, children.